Hello and welcome to our video lecture on Chapter 18, where we're going to discuss adaptive specific host defenses. And we're going to start off with our first section, 18.1, as an overview of specific adaptive immunity and what it involves. So our learning objectives here are to define memory, primary response, secondary response, and specificity, distinguish between humoral and cellular immunity, differentiate between antigens, epitopes, and haptens, and describe the structure and function of antibodies and distinguish between the different classes of antibodies. We'll start off with our clinical focus on this little infant known as Olivia, and she arrived in the ER with excessive crying, irritability, sensitivity to light, unusual lethargy, and vomiting. So the physician felt some swollen lymph nodes in Olivia's throat and armpits, and the area of the abdomen in the area where the spleen would be, which is our upper left quadrant, is swollen and tender. So we want to think to ourselves, what do these symptoms suggest? And what tests do you think the physician should order to try and diagnose the problem? So let's start off tackling that first objective and defining a few terms here. So we say the adaptive immune system is specific, and that means that we're able to target a specific pathogen, whether it's a specific influenza virus or a specific bacterial cell. And we can develop an immunological memory as we attack this pathogen, meaning that we're able to quickly respond to pathogens that we were previously exposed to. So when we are first exposed to a pathogen, let's say it's our first time ever getting the flu, we are going to have a primary immune response. And we see that it does take some time to build up our concentration of the antibodies here. And eventually, as we combat that illness, we are going to reduce the amount of antibodies. But notice how it doesn't completely go down to zero. We always have them kind of circulating. And then when we are exposed to, let's say, the same influenza virus again, we are going to have a secondary immune response where we're able to develop those antibodies much more quickly and combat the illness more quickly as well, having fewer symptoms, and then it is going to go back down. So essentially, our secondary immune response is going to be much stronger and faster versus the primary response. Now, our primary cells that are working within the adaptive immune system are our lymphocytes, or most of you better know them as your T cells and B cells. And they're going to arise from a common hematopoietic stem cell and work their way down that differentiation pathway. And you could see that pathway taking place over here. When we talk about our humoral immunity, it's going to involve our B cells. And the reason that this was called humoral immunity is because centuries ago, they believed that our body had these four humors within it, these four pools of fluid. And within them, they don't know at the time, but these B cells would secrete antibodies in which it would combat a illness. So this came to be our humoral immunity. Sometimes it is termed our antibody-mediated immunity. You could see that listed over here, in which these B cells are going to arise within our bone marrow and stay within the bone marrow to mature. And they can travel to other lymphoid organs to hang out there to see if they run into a pathogen. And once they're activated, they are going to differentiate into plasma cells and create antibodies that are specific to a pathogen. We also call these antibodies immunoglobulins. The other side of this is our T cells, which work within a cell-mediated immunity, and they're going to originate within our bone marrow and then travel over to the thymus, which is why they're called T cells, T for thymus, and also B for bone marrow, since they mature there for our B cells. But let's go back to our T cells here. They're going to mature in the thymus and then again can travel over to other lymphoid organs, such as, let's say, the spleen, to see if they run into any pathogens. And once they're activated, they become our central orchestrators of our immune systems. and can destroy infected cells. So what are we going to use in order to alert our immune system? Most of the time, they're going to be our antigens or immunogens. And these are similar to the pathogen-associated molecular patterns, or the PAMPs, that we talked about in chapter 17. But our antigens are going to be more unique to pathogens. Remember that our PAMPs are molecular structures that we find on numerous pathogens, but the antigens are going to be specific to that one type of pathogen. And our antigens are going to belong to a number of molecular classes. They could be carbohydrates, lipids, nucleic acids, proteins, or a combination of these things. And so if we want to think about the specific antigens that we will see on bacteria, we're going to think of the capsules, the cell walls, fimbriae, flagella, pili, extracellular toxins that they may secrete, or enzymes. And when they're viral, it could be related to the capsids, envelopes, and the spike structures. And what we see is that as the antigen has a more complex molecule, they are more effective. And so when we look at molecular compounds such as lipids and nucleic acids, we see that they are the least antigenic molecules. So what we'll see as a stronger response is if they're made up of proteins, because those are pretty complex. And so the way we go about combating these antigens is that our antibodies and T cells will interact with something called epitopes, which you could see pictured up over here. Now, epitopes are also known as antigenic determinants. So those terms are interchangeable. And essentially, we will have different antibodies that can bind to these different epitopes on the same antigen. So they've nicely color-coded this in the figure below, but you could see that the epitope that is shaped like a square has this antibody that is specifically made to attach to that. Whereas we have another antibody that has a different shape to its antigen binding site, which we'll learn about soon, attaching to the cone-shaped epitope. So it's kind of like an ID marker, but it's very specific. And these epitopes are always able to elicit an immune response. Now, another term which some people relate to epitopes is haptens. Haptens are free epitopes that are not a part of this 3D structure of this larger antigen. And so on their own, haptens are not going to be able to elicit an immune response, but they have been responsible for some allergic responses. Now, they can become a trigger for our immune system if they attach to larger carrier molecules, such as a protein. So a hapten plus protein is going to produce something known as a conjugate antigen. And like I said, this can cause an immune response. But we typically are going to see they are more related to allergic responses. In the previous slide, we talked a little bit about antibodies and an antigen binding site. So let's take a look at the structure of an antibody or immunoglobulin and what it really is. These are glycoproteins that we're going to find both in our blood and our tissue fluids. So the way that they're made up is that we have a heavy chain, which you could see in the purple over here. And these are our two largest chains, and they're going to be identical to one another. The other that we see in the green here is our light chain. So these are our smaller chains, and they'll also be identical to one another. Now, as these come together, we create two regions. Our, what I'm going to call FAB region, and the FAB here is standing for fragment of antigen binding, which we also call the arm of our Y-shaped antibody. And you could see at the very end here, we have a variable region. And we call this variable because it's going to have a high degree of variability between each antibody, because we are going to have that specificity for each epitope that we saw in the previous slide. And so we're going to call this area the antigen binding site. Now, when we bind to an antigen, it's going to allow the antibody to function in a way to combat the pathogen. And we're going to talk about those functions in the coming slides after we discuss the different classes of antibodies. The other region that gets created is this constant region. And just like the name says, it's going to allow this structure to stay constant as the trunk of our Y antibody shape. And majority of the constant region is made up of our FC region. So FC stands for fragment of crystallization, and it's where the complement factor binding will take place and also allow us to bind to phagocytic cells during something called antibody-mediated opsonization. And one more thing I want to point out here in the structure is that we do have this disulfide bond that is a covalent bond between the R groups found on our 2 cysteine amino acids. That would be a part of the Y here. Now let's talk about the different classes of antibodies. So we said that the constant region is going to determine its class or isotype, and that is going to be IgG, IgM, IgA, IgD, and IgE. So each of these are going to possess unique heavy chains designated by Greek letters, and you can see those listed over here. We've got gamma, mu, alpha, delta, and epsilon. So let's go through the next slides to see what is unique about each of these classes, starting with IgG, also known as our serum antibody. This is a monomer, meaning we've only got that Y shape to our antibody here, and this is the most abundant antibody in our human blood. It makes up about 80% of our total serum antibody, hence its little name. We are going to be able to penetrate tissue efficiently, and it's our only antibody that is able to cross the placental barrier in someone who is pregnant. So it provides passive immunity during fetal development in pregnancy. And it's the most versatile antibody class. And we could see some of its functioning over here. We're going to discuss what these terms mean in future slides. Now let's talk about IgM. We could see that we have a shape change here because it is a pentamer. So initially we are going to find this on our B cells as its monomeric membrane-bound form. And it is then going to be secreted and it can assemble into a pentamer that is bound by what's known as a J chain, which is a protein. This is our first antibody that we're going to produce in that primary response. So the first time that we are infected with a pathogen, and then also within the secondary immune responses. But we will, of course, produce it much faster in the secondary response. So we can use it as a valuable diagnostic marker when we are having an active or recent infection. Our third antibody is IgA, and this is also known as our secretory antibody. It is found as 13% within our serum, but the reason we really call it secretory is because we find it as the most abundant and common antibody in our mucus secretions. So we find it in areas like breast milk, tears, and saliva, and we can see it assemble into this dimeric form when our 2 monomers are joined together by a protein known as our secretory component. And you can see that highlighted in blue here. What it does is it traps pathogens within the mucus so it can later be eliminated from the body. Fourth, we have IgD, and this is the most like IgM. It is a membrane-bound monomer found on the surface of B cells. That's its commonality, and it's serving as an antigen-binding receptor. So we would say the B cell is down over here in the constant region, and then this variable region is open for the antigen. So it's not secreted by our B cells, and we only find trace amounts detected in our serum. And that really comes from the degradation of our old B cells, and it's releasing IgD as it degrades. Next, we have IgE. This is the least abundant antibody class in the serum, and it's most like IgG. We see it in its monomer form, similar to IgG, and it has a role in anti-parasitic defenses. The FC region over here is going to bind to cells like basophils and mast cells. So that would be the attachment down here to our cells, and the Fab region up here is going to interact with specific antigen epitopes, which cause the cells to release potent pro-inflammatory mediators. And we see this reaction as being central to allergic reactions, which, as you know, mast cells and basophils play an important role in. Now let's take a look at that closer relationship between the antigen and antibodies. We can see our antigen over here, and we've got two different types of antibodies attaching to them at their antigen binding site. So some of the functions that we can have take place now is neutralization, opsonization for phagocytosis, agglutination, complement activation, and antibody-dependent cell-mediated cytotoxicity. So let's visit each of these functions to understand what that really is. We'll start with neutralization, and we have certain antibodies, mainly IgG, IgM, and IgA, that are able to bind to the epitopes on the surface of their pathogen. So here we see an example of the virus and antibodies attaching to their spikes. Here's the diphtheria toxin in which the antibodies attach to, or this bacterial cell's flagella, right? And what it does is it's preventing their attachment to cells or preventing them from entering and infecting cells, thereby neutralizing their toxic effects or their ability to penetrate something such as our intestinal mucosal cells and causing destruction there. The next function of our antibodies is opsonization. And I always say this is kind of like putting a big red X or a target on the pathogen that we're trying to get rid of. So think of like when deforestation takes place and they're putting red X's on the trees that they need to chop down or whatever it is. That's what opsonization is. So what they do is they find the pathogen and the antibodies start coating it with their molecules, their bodies. These can include complement factors, C-reactive protein, and serum amyloid A. And what it does is it tells those phagocytes, hey, come on over here. And it allows that phagocyte to bind and ingest or engulf that pathogen so that it could go through the process of phagocytosis. So our pathogens that are our phagocytes that are able to do this are our macrophages, dendritic cells, and our neutrophils that have the receptors on their surfaces that recognize that Fc portion of, let's say, our IgG antibodies and can attach and engulf that pathogen. Another function of our antibodies is agglutination, or it's called aggregation. And this is one of my favorites because in the term agglutination is the word glue. So I think literally of gluing these pathogens together. It does this by cross-linking the pathogens by the antibody to create these large aggregates. So what I mean by that is that we are able to take those antigen binding sites and attach to two different epitopes on a pathogen. Now, when we look at IgG, we know that's a monomer, and we only have one of these Y shapes to it. So it has these two separate pathogen areas that it can attach to their epitopes. So they are able to form large aggregates, but what we see pictured over here is IgM at work, which is going to have 10 binding sites per molecule. So this is the most efficient antibody for agglutination. And you could see that the antibodies are attaching these different pathogens and bringing them together, again, creating a large aggregate. So as we get these clumps, it makes it easier for our kidneys and our spleen to filter them from our blood, and also makes it easier for our pathogens to come in and recognize these pathogens so that it can ingest it for destruction. And yet another function of antibodies is to allow for complement activation. By coating an antigen with an antibody, we can promote the inflammatory response and recruit our phagocytes and enhance phagocytosis by opsonization and kill our gram-negative bacteria using that MAC complex. There are those three different pathways that we've talked about before, but the classical is going to be the most efficient. What we see is an initial binding of IgG or IgM to the antigen, and that allows the recruitment of our C1 complex. And after that, we get that cascade, right? Where we're going to cleave certain complement proteins and allow enzymes to come in. And essentially, we're getting down to the end goal, which is creating that MAC complex, membrane attack complex, to create pores, allowing the fluid to move in, having the cells swell up, and eventually burst or lysis. The last function we're going to talk about is antibody-dependent cell-mediated cytotoxicity, also known as ADCC. Now, this enhances the killing of pathogens that are too large for our phagocytes to engulf. And this is best done by our natural killer cells, the NK cells, but can also involve macrophages and eosinophils. And the way they do this is first the antibody, here we see pictured IgG, is going to have that Fab region attached to the epitope or antigen on the pathogen. And then we see that the constant region, or the FC region here rather, is going to attach to our NK cell, or like we said, a macrophage or eosinophil, and then it's going to allow it to release these powerful cytotoxins such as perforin and granzymes that we've talked about, released from NK cells before, that kill the pathogen.